Hi there, this is Aaron Filler. I'm a neurosurgeon in Santa Monica, California, and I'm going to be taking you through the basic science of MRI, how DTI works, and then going into the details of diffusion MRI, how DTI was invented. We have a video section here that explains how DTI data is processed and how you can make a tractogram. We're going to then review the various clinical applications for DTI and tractography, and then talk about the newer field of connectomics. This basic science of MRI section lays out the fundamentals of the magnetic fields and the gradients and the radio frequency, what the protons are doing, as a background to being able to explain what's happening in diffusion MRI and how diffusion tensor imaging, or DTI, works. So the essence of MRI are spinning protons. These are the hydrogen atoms in H2O of water. And in a magnetic field, the protons spin, and they have a fixed speed determined by the strength of the magnetic field. If extra energy is pushed into the protons, they can't spin any faster, but they will start to precess, that is, to go off axis. So you can pump the energy in, and as you get more energy in, the black arrow here would go almost flat, parallel to the two lower black arrows. And then we'll see, if you click on the video in the second panel, that once you turn off the radio frequency stimulation, the proton gradually loses the energy that's been applied and comes back up to vertical. In order for this process to work, the energy that's sent in, the radio frequency energy, is tuned to the exact known frequency for these protons. Again, it's set by the magnetic field, 3 Tesla, 1.5 Tesla, and with the transmit antenna, we send the radio frequency resonant energy in, and then when we stop stimulating, as the decay takes place and the proton comes back up to vertical, that is giving off a signal which we can then record, demonstrating this process of decay. Now, some of the other technical MRI language we use has to do with pulses, a 90-degree pulse and a 180-degree pulse, and then the echo. So the spinning protons are first hit with enough energy to flip them on their side, and then as a group of them undergoes interactions with the atoms around them, the sharp peak, where they're all pointing the same direction, spreads out. We then give it a 180-degree pulse, and the same forces that spread apart the arrows bring them back together again. It's a strange concept. It's the spin echo concept. And we use this to refocus or bring the signal out. So a 90-degree pulse to flip the proton on its side, let it interact with the environment, flip it over, and then let the same interactions be reversed to get back to the signal we want to listen. But we're going to pick up some subtle differences along the way, despite the 180-degree pulse that does the correction step. This is just another representation of that. In A, the proton is vertical. In B, it's got the 90-degree pulse knock on its side. Some protons speed up, some slow down as they interact with other atoms around them, with iron in the environment. The peak spreads out. If they're all affected very uniformly, we give that 180-degree pulse in E there, and then they start coming back together again. They won't quite get perfectly realigned if there are asymmetric influences, tissue influences, and this is why the decay ends up being different. In different tissues, you get some turn out to be bright and some turn out to be dark, and gives you the contrast in an MRI image. An MRI scanner has a series of gradient coils. So this is in addition to the main magnetic field. There's X, Y, and Z coils that give magnetic gradient fields, which we'll say more about, that cross the person head to toe, right to left, front to back. And these are the essence of MR imaging. Here again is this concept that the magnetic field is strong on one side, for instance, on the right side of the person, and weak on the other side. So the strength of the magnetic field decreases as you move across the object, across the person and the imager. And this is important because, remember, the speed of spinning is determined on the strength of the field. So the field's stronger on the right, the spinning speed will be faster, and the field is weaker on the left, the spinning speed will be slower, so we can actually tune in with the frequency, the stimulation frequency, and the frequency coming back out as a return signal, or whether we're listening to something to the left or to the right based on the frequency. And here we see that again, one gradient going left to right, what we can call a frequency encoding gradient, a gradient going front to back, a phase encoding gradient, and one going head to foot, which is a slice selection gradient. And basically in MRI, we actually will tend to activate just one slice of tissue and then throw all the energy in at that frequency and then move to a different slice and then do that slice and then the next one. And then within each slice, we've got to lay out the X and the Y. So the Z is the long length of the person. We lay out the X and the Y with the two other gradients so that each location in the volume has a unique field strength. And here's that concept of the protons in a stronger magnetic field will spin faster. The ones in a weaker magnetic field will spin a little slower. And that gives us an addressable frequency that lets us locate each voxel or three-dimensional pixel in the image based on its frequency. And this is the difference between NMR, nuclear magnetic resonance spectroscopy, where a whole volume is applied a single magnetic field strength and as a single signal coming out, versus MRI, which was invented by Damadian and by Mansfield, which has the set of X, Y, and Z gradients, which give a unique field strength, a unique frequency to each voxel, and lets us to actually make an image by being able to tune into the different voxels. And I apologize for repetition, but here it is to see it again, a Z gradient, a Y gradient, an X gradient, and whether they're turned on all at once or in sequence, it ends up with a unique strength or unique frequency for each location, allowing us to make an image. Now we're going to move on to diffusion MRI, which works a little differently. What we've been talking about up till now has to do with T1 and T2, many of the routine types of MRI, which carried us from the mid-1970s into around 1990 or so, when Michael Mosley introduced the use of diffusion in MR imaging. First off, what is the diffusion we're talking about? The diffusion is the Brownian motion of water molecules, that is just heat energy, enthalpy, if you remember from physics, that causes water molecules to move around. And we classify the way the water molecules move in neural tissue and other tissues in the brain or any part of the body into either isotropic, they diffuse equally in all directions, versus anisotropic, they tend to diffuse directionally. And that is really something very striking and special about axons and neural tracts, is the water tends to diffuse along the length of the tract up and down more than it goes through the sides walls of the nerves, and it sort of makes sense. And so it actually becomes an important measure of the health of nerves. In isotropic substance being imaged, such as CSF, the water is freely diffusing in all directions. Whereas in an anisotropic tissue, such as the white matter tracts or peripheral nerve, water diffuses up and down along the axon. Now, it is true that in gray matter, in say the cortex, there ought to be directional diffusion, but the fact is that the neurons are so small, and the branches and the dendrites are so complex that you can't resolve it. But in a white matter tract, you have large bulk groups of axons traveling considerable distances in a similar direction, and you can pick up this directionality of diffusion. And that's fundamental to diffusion MRI. Now, the first application of this, DWI, which you hear about quite a bit in imaging, was the first way to really make stroke show up in MRI, because one of the things that happens in a stroke is very rapidly the pattern of diffusion changes in the ischemic and certainly in the stroked out or infarcted tissue, so that while the image still looks the same at a standard T1 or T2 MRI, the pattern of diffusion starts to change. And with DWI, we can detect this overall change in diffusion. We class them as restricted or unrestricted diffusion without talking about isotropy or direction, because it's a general assessment of the volume without looking into this directional question. The TI, diffusion tensor imaging, and a few other variants, diffusion spectral imaging, and it goes on and on these days, have to do with getting this directional information. Now I'm going to say a little bit more about what's going on in this slide, what we're depicting. And moving from this issue of just restricted, unrestricted, general diffusion to being able to monitor the direction, determine the actual direction in space in which an axon is oriented with the measurement process. So the idea of this is that if you direct a gradient across the tissue here, we have one called gradient axis perpendicular there on the left, you'll see that as the water diffuses, and we've shown this, the white is a strong gradient, gray, weak gradient on the left. So if the water is diffusing up and down along that nerve, it's staying in that stronger white gradient there. And as it diffuses, it stays in the same field strength, and so the proton spins remain at the same speed. Whereas in the isotropically diffusing tissue to the left there, the arrow is pointing in all direction. As the water bounces around, the water in a voxel starts, some of it moving towards a weaker field, some moving towards a stronger, they move in all directions, and the whole thing starts to lose phase coherence. The signal starts breaking apart, and a decay appears due to the fact that they're spreading around, whereas the protons in the nerve, where the gradient is parallel, are not affected by that decay, and they become brighter than the isotropic tissue. But now, different situation on the right of the slide, where we have the gradient oriented along the length of the nerve, so it's sort of white at one end of the nerve and dark at the other end. Now, the isotropic volume, it doesn't care which direction you have the gradient oriented. It has the same amount of decay due to diffusion. But with the nerve now, the opposite thing has happened. On the left, as the water diffuses, it stays in the same field strength. But now, when the gradient is turned parallel to the nerve, then the water is actually more rapidly moving either to the stronger or weaker gradient in a more purposeful way than in the isotropic tissue. So here, the nerve tends to go black, lose its signal faster. So when the gradient is perpendicular, the nerve stays bright. When it's parallel, the nerve goes dark. Isotropic tissue doesn't matter, and you begin to think, wait a second here, by orienting the external magnetic field gradient, I'm going to be able to detect which way the nerve is pointing, because if I crossways to the nerve or the neural tract, it'll stay bright, and if I'm parallel, it'll go dark. So instead of the T1 or T2 being from CSF or tissue, instead of the tissue, it's actually going to be determined, the brightness is going to be determined by the orientation of the neural tract relative to the orientation of my applied gradient. Now I'm going to introduce another concept here, which is how an MRI scanner is controlled, or how a designer or inventor goes about making MRIs do new things. So this is a pulse sequence, and it shows the sequence in which certain magnetic fields are turned on and off, the sequence in which certain radio frequency pulses are turned on, and how they interact with each other. And this is really how the detail of MRI is written out. And it was in one of these steps, the phase encoding, figure 11E, from this this patent filed in 1993, published in 96, that it showed how to be able to do a critical step in determining the orientation of nerves. And this has to do with the fact that although we can do X or Y, parallel or perpendicular, the neural tracts in the brain obviously follow very complex curved paths. So you can't just pick a direction like you would for the median nerve in the forearm. So the solution to determining which way nerves are going is to shoot the gradient in from a minimum of 6 different directions for each voxel, and then do a mathematical determination which calculates the orientation of the vector, essentially effective vector. It goes to the tensor math is one of the ways it's done to calculate the direction of the vector. So instead of just showing an intensity in the voxel, we're going to come up with a vector that shows the orientation in space of the dominant group of axons in the voxel, and the length of the vector will encode whether it's highly anisotropic or marginally or non-isotropic, anisotropic at all. And the key thing in the invention of DTI was to figure out how to be able to shoot in these diffusion magnetic field gradients without wrecking the X, Y, and Z pattern that was essential for imaging. And the solution there is in that line 11E, which was to take a gradient, run it in, do the 180-degree pulse, which we had fixing noise before, and then hit the gradient oppositely again. And between those 3 activities, it became possible to remove the interactions between the diffusion gradients and the location gradients, made it possible to shoot the gradients in from any angle, and in modern imagers, 30, 60, 200 angles. All that's done really fast, and none of it degrades the image due to this critical breakthrough of understanding how to mix diffusion gradients off angle with X, Y, and Z gradients. So we send the gradients in from multiple directions, at least 6 directions. We do the calculation. If the water is moving, if the water is diffusing isotropically, we get the sphere. If it's diffusing anisotropically, we get this sort of ovoid or ellipsoid shape, which is the tensor pattern. And then we can get the orientation of this within the voxel, which way are the axons pointing. The mathematical methods for calculating this directional question are numerous. They range from complex vector methods to tensor methods, and the advantage of the vector methods is that a tensor method can only show one direction in a voxel. And so this is a bit of a problem because in any given voxel, 2 axonal pathways may be passing through each other, they may be splitting from each other, they may be joining. So with a tensor analysis, you only get one answer per voxel. And if there's 2 axons, tracts passing through each other, it may incorrectly say there's no axons at all because it can't pick up a uniform direction. So the tensor method gets you one direction, and the various vector methods can often calculate multiple directions in a given voxel. With the tensor method, we get this calculation of an ellipsoid with the long axis and the 2 shorter axes. And you hear of these bandied about different ways that analysis is done, the lambda 1, lambda 2, lambda 3, mean diffusivity, radial diffusivity. The fraction isotropy is a summary number, FA, fraction anisotropy, wherein if it's highly anisotropic, it'll be nearly 1.0, and if it's relatively isotropic like CSF, it'll be nearly 0. And it's substantially captured by the long axis of the ellipsoid there. What you see in C is the concept of fiber tracking. What you want to do is have an algorithm that you start off someplace in the anatomy based on the known anatomy or a functional MRI, and you want the algorithm to follow the direction of the axons in your voxel, and then look at the next voxel or surrounding voxels and determine if there's a neighbor where the tract is going in the same direction. If so, it draws the line, then looks at the next voxel, looks at the next voxel, and goes on. And it's a surprising thing, but this actually maps out the known tracts of the brain. And we did it for the very first time in early 1992, and it worked. We got a corticospinal tract. This is really the basis for fiber tracking methods. There are, again, numerous different fiber tracking algorithms that have benefits. We'll talk about the differences among them. But this is the very basic essence of DTI and tractography, which is then the next what you do with the DTI data to look at higher order structures such as the tracts of the brain. And this slide just depicts this issue that we have the X, Y, and Z gradients, and then we're throwing in a diffusion gradient at a different angle. It causes what's called a cross term that distorts the image. And this is the problem that was solved by splitting the gradient pulse, which made it possible to shoot the diffusion gradients in from multiple directions, up to an infinite number of directions, really, and then just use mathematical processes on a voxel-by-voxel basis to calculate the direction or directions of axons passing through the voxel. And then we took this 180-degree inversion concept from spin echo imaging, which goes back to the 1950s, and used this 180-degree flipping technique to correct out the interactions, the destructive cross terms between the diffusion gradient and the locational gradients, the X, Y, Z. And here, for instance, is a standard pulse sequence setup. We have the GSS, GPE, GFE. These are the slice select gradient, the phase encode gradient, and the frequency encode gradient, which would typically just be set up early on. And then we get the echo later. But what we have to do is to split one of these and put it on either side of that 180-degree pulse. These pulse sequences are run over and over again. If you listen to an MRI, you hear that knock, 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 knock. That's each time one of the sequences run. These may be less than a second, and they're repeated as they gradually step through the volume, through the various slices and the various phase encode settings. And here again, we see the specialized sequence for diffusion anisotropy imaging, where we take the phase encode gradient, split it, have it go with different polarities on either side of the 180-degree inversion pulse. You see at the top, the 180-degree radiofrequency pulse. And at the bottom, we're showing the diffusion sensitizing gradients. And the way these work is it's called a Steckel-Tanner pulse, S-T-E-J-S-K-A-L, and then Tanner for the two scientists. We apply another gradient for diffusion, turn it off, do the 180-degree pulse, turn it on again. And if the water hasn't moved, in the diffusion gradient, it'll come out the same. If it's in different positions between the two diffusion gradient pulses, it'll show the decay. And this process of using split and inverted pulses to allow the diffusion gradient to cross through the location gradients without ruining the image is pretty much as we initially proposed this and published it in 1992, and is used up to the current times with various enhancements and further adjustments in what we call single-shot diffusion imaging. Now, you may be wondering, if we have to do the image maybe 20 or 30 or 100 times, why doesn't one of these diffusion scans go on forever? And the answer is that the underlying type of sequence used is what's called an echoplanar sequence. These are really fast, and it's able to do that large number of images fairly quickly without being much longer than a routine 5 to 10 minute MRI sequence. We still have to do the splits on the phasing code, for instance, gradients, in order to get them through without being interfered with by the diffusion gradients that are applied at the non-orthogonal or non-Cartesian angles. And this is the very first tractogram that was done, and you can see that we were able to get the orientations for the corticospinal tract as a curving structure, right and left side, and then use a connected voxel algorithm, which was similar to what had been developed previously for doing MR angiography of the carotid artery, to follow the tract and give it contrast based on the shared directionality. Now, fortunately, it turns out that modern tractograms are really very, very good data. Having a good tractogram greatly improves the safety of tumor resections, because you can really see exactly where the tracts are to avoid them. Yes, there are problems, there are challenges, there are optimizations that go on, but fundamentally, this is a very helpful technique which is continually improved and optimized. Here, for instance, we're seeing the superior longitudinal fasciculus, SLF, and then more anteriorly in the brain, the UF is the uncinate fasciculus, which conducts fibers from the amygdala up to the frontal lobe. So when it's impaired, you get a flattening of affect. And then it's closely intertwined with the inferior fronto-occipital fasciculus, the IFOF, which is involved in visually based memory formation and recall tasks. So, for instance, impairing that interferes with people's ability to form memories of new faces and names of people they've met the past few weeks. And you can actually see the structure that does this kind of elemental part of neural function. In tremendous degree now, we're able to see these and see when they're injured on a function-by-function basis in the brain. And the findings for the tracts are very robust. Here you see the same patient imaged in two very different scanners with different algorithms, and we have the uncinate fasciculus right and left side similarly decreased, despite it being two different images with two different algorithms. Now, the impact of these different tractographic techniques is significant. The classic is DTI using Tensor and a deterministic algorithm that marches from voxel to voxel. Another alternative is a probabilistic algorithm using DTI for the original processing. And here you're letting the computer work really hard to figure out possible alternates to the path from voxel to voxel. And this is really important, for instance, in the optic radiation, where the tracts come out from the geniculate and then run a little bit forward and then take this Myers loop turn to go back towards the calcarine cortex. And at that sharp turn, the deterministic algorithm may lose the tract, may not follow it around the curve, or probabilistic will do a better job. And on the right, we see CSD instead of DTI, which is constrained spherical deconvolution. And so this is gives you even more tracts. You see, we're losing a lot of the tracts in the arm and face in the two methods on the left, because you have crossing tracks that are confusing it in the algorithm. Constrained spherical deconvolution is one of these that essentially takes the voxel, and instead of asking, well, where's the one vector going here, where's the one main axis of the tensor, it treats it as like a multi-tessellated dodecahedron, like a complex igloo with a lot of surfaces like you might see for a radar system, and asks face-by-face basis on that multifaceted sphere, are there any major axes going through this face or that face, and you can come up with multiple directions and then use the probabilistic algorithm to find different ways, the optimal way to put it all together. This does get slow, even for a supercomputer. So there's always a balance between optimization, the forest blocking the trees. You can't see the forest for the trees, and the time of processing. You see is a scan here, 48 little image slices starting from the base of the skull on up through the brain to the very top of the head. Here are a couple of the image slices. They're exact same location. This is from our subject patient. This is a part of the pyramidal tract. It's bright on this image, dark on this image. That's showing it's anisotropic. So we're beginning to be able to get access to a selected structure, and eventually we'll get it to the point where we get a complete picture of just that selected structure, and we can make everything else disappear. And with different orientations of the gradient direction, each of these slices will look different depending on the direction of the gradient, because they've got anisotropic tissues in part. So we're getting a series of sets of images to be able to do the analysis. Vector processing collects up all the different versions for each slice and calculates and comes up with this summary. So for each location in the brain, each voxel can calculate a direction, and the color is just used to label which direction it's pointing. And the length of the vectors, how much anisotropy, so the bright areas are anisotropic, the gray matter is not lit up because it's not anisotropic, it's isotropic. But this is what you get, is a full set of the data for the whole brain. But this is not tractography yet. This is the result of the vector processing. So in this patient that we're working on, I've got the DTI vector processing, complete data collected, vector processed already. So I'm going to go down in the image here to find that cerebral peduncle structure there, and then I place my crosshair on it, and then have the software give me a volume of interest box. I'm going to pull it in tight on the anatomical structure I've selected. Here I've chosen to do the generation of the pyramidal tract for me. Pull it in close here, and then I will give the software some parameters here. Set it up to be a seed. So now I've got my region of interest or volume of interest in place on the cerebral peduncle on the right side. Seed in 3 dimensions. I'm going to set some fraction isotropy parameters here, and then we can run this, and there it goes. So to take a look, now we have a pyramidal tract. Blow it up here. There it is. So now I have selected my pyramidal tract because I selected a structure along the way. I can, just for demonstration, pick out an area over here where the vertical spinal tract would come to the surface that we would see from the fMRI if it was performed here. And having set up that second location, which would be what the fMRI would show us, we add that as a seed and get a little more dense or effective tractography from that. And now I can pretty much make the rest of this all disappear, and we can just look at the pyramidal tract mostly in isolation. So we now have a nice view of a data representation, virtual representation of our selected structure, which is a pyramidal tract. In this scan, I have them marked with arrows, a few spots that are taped onto the skull, a couple around the ear, behind the ear, back of the head. So up here we are looking through a microscope at the patient's brain, and the software then is able to take the virtual pyramidal tract and project it in a heads-up view, give it the correct, show that it shows its position, orientation, and shape in the region, which is in the patient's head, in the patient's brain at the time of surgery. So now we want to talk about some of the clinical applications of DTI. And it first should be clear that the amount of information about DTI is very large. There's more than 20,000 peer-reviewed publications. It has a big role in a number of areas of neuroscience and neurology, for instance. Someone in their 60s is starting to have trouble remembering words and run into other memory issues. They want to know, is this just the routine memory loss of aging, or are they getting Alzheimer's and need to wind up their affairs? And these have very different appearances as to the pattern of anisotropy in a DTI image, and so you can really sort these things out. Now, for neurosurgeons, one of the most important applications has been guidance during tumor sections, both to minimize injuries to critical tracts, but also to maximize the amount of tumor resection, so you can see just how close you can get to a tract. And again, these are always undergoing optimization and improvement with the elapse of time and a tremendous amount of scientific effort, but has had a huge impact. Starting in 2007, papers were published showing a decrease in mortality during major tumor sections, so as great as 40% decrease in morbidity, such as paralysis, muteness, blindness, by as much as 60% when you're able to have tractographic guidance. And this is simply because, as neurosurgeons will know, white matter doesn't allow you to distinguish areas that are tolerant to resection, areas that are not. You can remove some frontal lobe or temporal lobe as needed without necessarily having a dramatic impact, but you can take a tiny amount of pyramidal tract to have a permanent paralysis. And obviously, being able to see the difference while you're operating has had a huge impact. This goes for any progress through the brain, whether you're doing finding the best way to approach intracerebral hematoma. But there are a number of other very interesting areas. One of the biggest effects has been in the area of evaluation of head injury, because the fact that we can identify individual structures and see dropout due to fractional anisotropy losses has allowed us to, for instance, identify exactly why people get a memory formation failure. What is photophobia? How do people get pain from bright lights? What is causing central vertigo, that is vertigo not due to a middle ear problem? All of these are types of things that are now reliably evaluated by DTI in head injury and concussion. Also, hypoxia, where you have watershed regions, such as around the superior longitudinal fasciculus, giving dropout and fractional anisotropy, and evaluation of tremor. Is it Parkinson's? Looking at the substantia nigra as to fractional anisotropy to determine are there losses that will indicate a particular type of a cause of tremor. Here, simply enough, is an example of using tractography to plan an approach to an intracerebral hematoma. What's the safe way in to avoid the critical tracts? And here's a nice demonstration from Memorial Sloan Kettering of using fMRI, bold, blood oxygen level dependent imaging to identify Broca's area and Wernicke's area in an fMRI session, and then continuing the session to collect the DTI data, then using the Broca's area and the Wernicke's area as the two seeds to form the image of the left arcuate fasciculus. Here, where it's distorted out of its normal position by a tumor, and then it allows you to come in and resect the tumor without resecting the arcuate fasciculus. Now, this is an image to do with a process of evaluating head injury. And if you notice, For instance, in the lower left of the 6 panels, a small white circle over the blue structure. And this is a volume of interest. So what's going on here is that it is possible to lay down a series of these selected volumes of interest and measure the fractional isotropy and compare left and right side and compare to normal standards. So, for instance, in the textbook chapter, we provide normal standards for fractional isotropy of various parts of the brain based on normal individuals assessed under IRB with appropriate background data for a number of brain structures so that we can understand when there's a statistically significant difference right to left or a statistically significant difference between the fractional isotropy of a structure you're measuring and the fractional isotropy of a standard normal in order to detect the presence of pathology after head injury. And many neuroradiologists are using software that can only look at the corpus callosum, but modern analysis software can allow detailed analysis of numerous areas of the brain. You have ability to look at the supracallosal cingulum to see if there's a problem with depression and anxiety being triggered, look at the hippocampal cingulum to look for attention, look into the fornix for memory loss. All of that stuff can be usefully studied, and there's great data showing the utility of this in understanding the persistent post-concussive symptoms or progressive post-concussive symptoms that can occur after trauma. This is just a further example of some of this process of selecting different volumes of interest, the AF, the arcuate fasciculus, the AG, angular gyrus, which would affect word finding and calculation ability, the UF, uncinate fasciculus, and inferior frontooccipital fasciculus, with the UF affecting flattening of affect, loss of emotional drive, and the inferior frontooccipital fasciculus affecting visually based memory formation and recall tasks. In C, we see the medial lemniscus. And so, although the medial lemniscus is a general sensory tract, it is used as a marker for some injuries to the midbrain tracts, which can affect convergence, accommodation, and be involved in photophobia. And then we're seeing on the bottom right there the genu of the corpus callosum, the splenium, the mid-body, and then the pillars of the fornix and the body of the fornix. Each of these are typically feasible for routine measurement and statistical evaluation. Similarly, the tractography gets better and better, and more particular here, we're seeing a very nice demonstration of the fornix and proximal limbic system, the hippocampal cingulum involved in attention, the fornix, new memory formation, supracallosal cingulum involved in anxiety and depression, and being able to target those with transcranial magnetic stimulation, for instance, for restorative treatments, and that is being able to look at a tract and follow connecting tracts that take you up to the cortical surface where TMS can access, and looking for underlying FA fractional isotropy dropouts that can demonstrate injury. By combining fMRI bold studies with tractographic analysis, there's a relentless progress to identify in a finer detail exactly which structures are involved and exactly which functions and behaviors and what happens when these get injured, whether it's executive functions, various language functions, different types of memory, immediate memory affected by the thalamo-prefrontal tracts, verbal working memory versus new memory formation, fornix structures, and some of the other structures involved in longer-term memory, which are still complex and being progressively better understood with the elapse of time. In a formal analysis of the fornix after head injury with the person complaining of persistent difficulty forming new memories, you can come along shortly after the accident and a year later and 2 years later and measure the fractional isotropy in the fornix structures and get the statistics out, look at the standard deviations, see are the two standard deviations different right to left or from the normal means, and really get a very fine level of analysis. And this is compared to a routine neuroradiology evaluation where you sort of look at the brain and say there's swelling here, there's a hematoma, but this is really, really bringing together all the details of classic neuroscience into this structural and functional world made possible with the DTI and tractography. The issue with new memory formation is that we have a tremendous amount of sensory information coming in all the time. We don't record everything, but the temporal lobe and the hippocampus attaches salience or degrees of interest, and then sends the signal up through the fornix, but it's got to go up and curve around forward and come down to the anterior septal nucleus anteriorly to trigger the memory formation. And when it comes up out of solid tissue and then travels as a thin structure through the ventricle, it's susceptible to mechanical forces such as a rotatory impact or a side impact in motor vehicle accidents. So you'll get a bruise in the fornix as it transitions out of solid tissue into the liquid space of the ventricle. And this is the way in which you can identify or distinguish a mechanical injury versus a degenerative pattern of memory loss. The whole idea of photophobia is puzzling. Like, why would bright lights give the person a sense of discomfort or pain? But this is a very common complaint after a head injury. And, we can now look at the fact that patients with this symptom very often have a low fraction isotropy in the medial lemniscus. So these are midbrain injuries that we can understand in terms of the way these different midbrain pathways interact. Similarly, convergence, accommodation, and other eye movement disorders such as gaze-related vertigo have their triggers here. And the issue is to do with the fact that you have the cerebrum, which is large and massive, and you have the cerebellum, which is also relatively massive. And they may move differentially in an impact, causing twisting on a small structure like the midbrain that joins the two. And this is why these are common problems after head injury. So you have to think of the biomechanics and then be able to look and actually see, do people have these abnormalities that match with the symptoms, and how real are the symptoms? And you can often find the pathology that underlies it. This is just, again, showing the use of the medial lemniscus as a marker for injuries to some of these very small pathways, which are difficult to sort out, but within the range of capabilities for advanced DTI into the future, with 7 Tesla scanners and 9 Tesla scanners and higher numbers of acquisitions and better mathematical methods of analysis. Here, for instance, you have a person who's fallen back and hit the back of their head and is complaining of severe vertigo, triggered moving their arm. They experienced vertigo, so essentially it's a central vertigo. And what you see if you look at the middle cerebral peduncle is the very dramatic difference in size between the left and right side. There's an injury in the cerebellar peduncle, and that kind of mismatch will trigger vertigo, oscillopsia, impairment of smooth movements, and some auditory dysfunctions. And what we're looking at on the right is medial lemniscus. It's a little harder to identify if you're not used to looking at tractograms, but you can come to be quite experienced with looking at these with some practice. And this just shows the fractional isotropy measure in the middle cerebral peduncle on the left side image looking for vertigo, and in the medial lemniscus on the right looking for some of the eye movement and photophobia issues. As mentioned earlier, you can get hypoxia effects in watershed areas, and then use the fractional isotropy and tractography to identify the stigmata of hypoxic injury. And here, we're seeing it in missing portions of the superior longitudinal fasciculus, which is one of those areas often affected by hypoxia. And I mentioned earlier about Parkinson's, and we measure different zones of the substantia nigra for fractional isotropy thresholds. And of course, there's controversy about what these should be and how reliable this is, and this is ongoing work. But it has shown an approach to really analyzing this type of disorder in very fine detail. Finally, I want to talk about connectomics, which is another big advance in DTI. And this is a project like the Human Genome Project, the Human Connectome Project, to map all the connections of the brain. And this is just coming into clinical use, 2021, after publication of about 4,000 peer-reviewed publications in this field. This project involved a number of institutions around the country and around the world collecting large amounts of detailed data. And this is one typical plan from WashU St. Louis and University of Minnesota, where they were indicating how many subjects they used using high-field MRI, MEG, as well as fMRI, to try to assemble large databases of a variety of normals and a variety of people with different types of pathological conditions, whether they're emotional disorders, degenerative disorders, traumatic disorders, so that you can use an artificial intelligence methodology to examine a patient's brain and see how it fits into known patterns of damage and dysfunction or other types of pathology. This type of tractogram gives you an idea of what happens as you go up on the number of directions. You can keep going up on the spatial resolution, number of directions of diffusion, and break the data down to finer detail using, Hardy, HAR, DI, high angular resolution, diffusion imaging, vector methods, non-tensor methods, to be able to see crossing fibers and not lose tracks through methodology, as happens with some older tensor methods. This also gives us a new view of the brain, the connectogram, which can either be mapped into the brain anatomy or set up in a graph like this. This is from Dr. Arimi and others at the UCLA. And there are other versions of this, but allow you to lay out the connections of the brain to understand pathology, and to lay out the pathway for determining artificial intelligence analyses of the data. Another part of this effort is to rethink the parcellation of the cortical surface. A lot of this stuff goes back more than 100 years in terms of how many different separate areas of the brain surface, the cerebral cortex, should be identified. And by pushing that up to 300 or 400 locations rather than 80 or 90, and then using fMRI bold to verify these, and high resolution tractography, you can identify these numerous expected tracks from the connectogram to see where they're disordered, distorted, missing tracks, and use artificial intelligence to be able to take some fMRI data and detailed DTI data to get a tremendous amount of detail about the expected injuries, and to follow them with serially over time as to improvement or deterioration.